Okay, so let's see. Okay, so are you ready? Okay, so in that case, I will start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. And welcome, and thank you for joining us, the 30th seminar. I cannot believe it, already 30 times, and I'm doing this one. And also I call it University of Maryland Day, because we have the pioneer speaker from that university. Tomorrow is the Earth Day that promotes the awareness for the health of our environment and Earth, of course. And this virtual seminar would have many limitations, but it can bring together scientists and engineers who cannot travel easily due to many reasons, including COVID and long travel time or family commitment. In the future, virtual seminars would be an alternative or sustainable way to reduce carbon emission, although I personally prefer human interactions in person in real conference. In fact, I met today's pioneer speaker, Bill, 2 weeks ago in Portugal, and this trip is part of my 4-month trips in this year, and we had really great time there. And come on, this is Portugal, so we should have great time, and we cannot replace in-person conferences with virtual ones. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Bill Bentley. He is a distinguished chair in engineering and distinguished university professor at University of Maryland, and also fellow of many organizations, including AAAS, ACS, American Chemical Society, AIMBE, American Academy of Microbiology. In fact, he doesn't need any introduction. So I'll stop here to give more time to him. So Professor Bentley, the podium is all yours now. And thank you so much for your time and excitement to support this seminar series. Thank you.
Thank you. Can you hear me?
Yes.
Okay. Yeah, wonderful. Thanks so much, Tae-sok, for organizing this spectacular session and endeavor. It's wonderful that you bring all of these great young folks to the fore and give them an opportunity. I have 3 slides very quickly. I think everybody that's watching recognizes that synthetic biology, metabolic engineering have been highly successful in generating products from engineered cells. And that's sort of indicated by this genetically engineered cell and taking substrates and making high value products, whether it's artemisinin or a precursor to polymers, et cetera. The thing that makes this work is that we put these cells, a colonal population, into a bioreactor and allow those cells to just sort of do what they're programmed to do, and we don't disturb them at all, other than control their pH, their dissolved oxygen, their temperature, nutrient profiles, et cetera. Those are all contained and maintained in a very sort of static or at least optimal process. Biology, on the other hand, recognizes all sorts of cues. And so most cells live in this sort of lower right environment where they respond to molecular cues, physical cues, mechanical cues, etc. And those would all distract the cells otherwise that were put in a bioreactor from making a product. And so one of the things that we've thought about in our lab many years ago was to think about the cells as the product of synthetic biology, not necessarily the products they make, because cells can do some advanced things. And so if we put a cell, for example, into the GI tract of a human, maybe that could do something that's far more advanced than make a product. And so hopefully I can, let's see if I can change. Yeah, here. So many years ago, this is, I think, 2011 or something like that, we had been talking about in our group the concept of having cells be sort of bacterial dirigibles. So that's dirigible is like the blimp, as you've seen here. What are the things that this could do? Well, so it has motility, so they could go somewhere. It has a control module that allows it to actually function. It could dock somewhere, and then it could take action, which is indicated there. And so this concept was batted around in our group for many years, and we finally made some progress on this. And so I'll indicate that by some of the people that you might recognize that were here in our group many years ago. These are papers that they subsequently did later, not necessarily in our group, with the exception of Shen-Chen Wu, who's a faculty member in Taiwan. But basically, the idea here was to engineer cells that would take an advanced function and do something interesting and beneficial. So John March, who's chair of biological engineering at Cornell, developed a couple of different strategies. One to cure diabetes in a rat. Matthew Chang, who's the advisor of In-Young, who's going to speak, did some work where he engineered bacterial cells to recognize Pseudomonas aeruginosa, and her actually work is sort of far exceeds what happened in the original paper. And then down here, Shen-Chen Wu, he wanted to do something more like I described, where docking. And so in this case, he decided to put these little nanofactories onto a cancer cell. And because cancer cells have more EGFR on their outer surface, these cells would swim there and then recognize autoinducer that was made by these by the surface appendages on these cancer cells, and then recognize that and then deliver a therapeutic, ideally. So that concept of having engineering cells to do something beyond make the product, go somewhere, take an action, that was at the heart of the discussion in our group many, many years ago. And fortunately, we have In-Young here to describe her work that takes that much further than we ever did. So I'll end there and be happy to turn it back over to Tae-Sa.
Oh my goodness, thank you so much. I mean, you are truly visionary because you influence a lot of people, including Matthew. And actually, Matthew's MSB paper, the pathogen killing paper, is actually I wanted to pursue that idea even before I start. So he schooled me even before I start my own lab. So that is the visionary kind of the influence you have. I mean, thank you so much for your impact to global community. Your influence and your impact will be forever, I guess. I mean, many generations down the road. So thank you so much, and then thank you for your time. And I love your visionary talk. Thank you so much.
Thank you.
So yeah. So of course, I'm now the main speaker of today with a little bit longer introduction. So Dr. In-Young Hwang is a research professor at the Department of Biochemistry, National University of Singapore, the best university in Singapore, and then of course one of the best university in the world. And she is leading therapeutic cell and mammalian synthetic biology research at the Synthetic Biology for Clinical and Technological Innovation, so-called SynBYSS. In Young used to work with the Professor Matthew Chang at NUS and still kind of interact with him a lot. And Matthew was the Professor Bentley's lab alum, as we kind of already kind of mentioned. So Young is the Professor Bentley's academic granddaughter, and as you can see, how much impact Bill actually having right now. Prior to joining NUS, Young has received her PhD degree from the University of Auckland, New Zealand. So she's kind of traveling a lot of places in the field of cancer biology and therapeutics. So probably this is some kind of natural transition from therapeutic area, cancer area to microbiome, I believe. She has previously demonstrated how microbes can be programmed to execute antimicrobial activity against human pathogen with high degree of specificity. Her research currently focuses on understanding and reprogramming the functionality of microbiomes for human health and therapy. Most importantly, she is the first Korean speaker for this seminar series as a rising star, and the second Korean speaker after Professor Sang-Yeop Lee at KAIST, and she gave the brilliant, pioneering or inspiring talk early this seminar series. And especially, I thank Young for speaking today here, because her time right now is, I guess, 11 p.m., I guess, and she will be around with us until 1 a.m., I guess, right? So Young,
yeah,
please take it away. Thank you so much.
Thank you, Professor Moon, and thank you, Professor Bentley, for a really, really good introduction of the talk that's coming ahead. I'd like to share my screen first. Okay, can I just double check that I'm on the right? Okay,
yep.
Thank you. I'll just put that into laser. So I think this first slide is pretty explanatory. So good morning in those in US, and as mentioned in Asian context, good night and pretty much good night. Good evening. So today I'd like to share some of our work in our effort in engineering microbes, which are essentially intended to rewire some of the host microbiome interactions, and in hopes of having therapeutic effect, and hopefully put the microbes in the limelight as next generation medicines and with its potentials. So I'm in Asia, as has been introduced, but in a tiny island of Singapore, a lot of you may not know where exactly they might be. So NUS, SynCity, is based in Singapore, and it's a tiny little dot above the equator. It's a small island in Southeast Asia, and it's just literally 1 degree above the equator. You would need to zoom in several times to see it on the world map. And when you do, it'll be something roughly like this. So it's actually at the tip of Malaysia Peninsula, and that's Hong Kong. And it is perhaps one of the most densely populated and urbanized city in the world, with many iconic landmarks that you may or may not recognize. So this is what we call Gardens by the Bay. And this statue is called Merlion, which is iconic symbol of Singapore, overlooking the city hall and the river. And across the river, then there's this building called Marina Bay Sands, which is identifiable by these 3 pillars that supports a ship-like structure, and it has an infinity pool in it. Or for some of you, a grand tour of the island may have been introduced to you in the film Crazy Rich Asians. And a bit of a disclaimer, not all Singaporeans are all that rich, unfortunately. Due to multicultural society that you can find in Singapore, which comprises people of Chinese origin, Malay, and Indian living all together. And it has English as one of its 4 official languages. Yes, 4. Because of that, Singapore can be considered as the Asia for beginners, because English is spoken throughout the country, so you can communicate, as well as getting exposure to the rich Chinese, Malay, Indian heritage as well. So that's a bit of background to Singapore, but don't let the size of the island fool you, or reflect that the scientific rigor that is happening in this country. So supported by many agencies and funding agency to promote and drive synthetic biology in Singapore, we have a good research center and many institutes that do research in synthetic biology. And we have many international collaborations, and it goes all the way to University of Maryland and various other places. We are well connected to synthetic biology communities in the US as well as Europe. So furthermore, we are also part of an Asian community of synthetic biology called ASPA, or Asian Synthetic Biology Association, which kickstarted in 2018. We had a break in the conference in 2020, which went virtual, but again, we hope that in-person conference will resume soon. So that's kind of the context of the NUS and SynCity. Here at NUS, I'm also part of SynCity, a research center of excellence in synthetic biology research. Our mission statement is written here, but at its core, we strive to be known for and deliver innovation and technological transfer capabilities research in synthetic biology. So it's actually quite grand. With this idea in mind, our research center has the research directions in 6 clusters, ranging from biomanufacturing, synthetic genomics, environmental remediation, genetic tools, sensors, and therapeutic cells. Today, we are very fortunate to have support from both public and private sector to support and promote these research directions here in Singapore. But today I will be sharing some of our work in the therapeutic cells and our efforts in developing towards live biotherapeutics. So that's pretty much the intro I have, and I'll get into the science of it. So as we all know, and also reiterated by many of the talks that has human microbiome as its topic, our body houses a number of microbes. Pretty much every corner of our body contains a large number of microbes that are living together and evolving together with us, our body. So because it's already there, one of the questions that our lab and also inherited from many different labs is that, can we give new functions to the microbes so that they can function as a potential therapeutics? So this is a grand question and the quest that we set ourselves on, and it's a very long-term vision that we have. So we aspire to develop a platform technologies where microbes can be reprogrammed or engineered to manage health and disease conditions. So if we can allow microbes the ability to detect an array of health and disease conditions, like those that occur in infection, then during inflammation happens, or cancer or metabolic disorders. We can use microbes then for detecting these disease and the diagnostic measure. In addition, if we can give microbes an ability to insert cellular functions that lead to therapeutic efficacy, such as basically the reverse anti-infection, anti-inflammation, anti-cancer, anti-metabolic disruptions, to have antimicrobial activity and immunity enhancement and so on. We can use microbes for disease management and hopefully this term as life therapeutics. So if you look into how of these a little bit further, so if you look into what kind of biomarkers there. So zooming into biomarkers and therapeutic molecules we can employ, and we can take advantage of the fact that we're using living microbes to do this. You can deduce that there are a range of biomarker molecules that are produced by host metabolites, or even external physiological signals such as pH, CO2, that microbes can use as a signal to detect. And if you couple them with an array of therapeutic molecules, such as antimicrobial peptides or metabolites or enzymes that microbes can produce to coordinate some output function, that could actually lead to our therapy. And then there's also a cellular phenotype of our microbe that we can enhance, such as their cellular adherence to promote colonization and motility, and how it's optimized to target and direct the therapeutic functions, and can be also employed together with therapeutic molecules. So this is this is also eloquently presented by Bill earlier on. So this is actually a sort of continuation in our aspiration to our research. And yes, the MSD paper in 2011, and this is now more than a decade-old paper. But I like to mention this because this is actually our origin story, where our idea or the concept actually take form and actually got tested. So when we first conceived to have an E. coli to detect a human pathogen and deploy a pathogen-specific peptide, thereby generating a highly specific pathogen-killing microbe, was pursued by selecting Pseudomonas aeruginosa as our testbed human pathogen. We chose this because not only is a notorious nosocomial infection agent, but it secretes a unique quorum sensing molecule that they use to communicate with each other. And because it's a communication purpose, it also has a responsive regulator called SR that is already capable of detecting this quorum sensing molecule. So we borrowed this design from the Pseudomonas and introduced to E. coli. So this enabled now E. coli to detect the molecule, the quorum sensing molecule secreted by the Pseudomonas. So then we coupled this with antimicrobial peptide that has specific targeting ability. And our search for such peptide resulted in identifying a pyroxene S5, which is a bacteriocene that was produced actually by other microbes to target Pseudomonas, and which was relatively under-characterized at that time. So our lab took the sequence, expressed and characterized its activity against a panel of Pseudomonas and realized it's highly specific to the Pseudomonas owing to its putative receptor binding domain. So it had a domain that we hypothesized that it recognizes a receptor of a surface protein of the Pseudomonas and subsequently insert a core-forming domain to target the Pseudomonas aeruginosa. So, but because that recognition is absent in E. coli, it can be perfectly safely expressed and accumulated in E. coli. So now that we have sensing and now we have killing peptide, we looked into a release mechanism where we employed the most simple and effective way, cell lysis. So we actually expressed the lysis protein to release the expressed antimicrobial peptide, and therefore we were able to generate an E. coli that is able to sense and kill the Pseudomonas. Of course, now this sensing and killing or sensing and responding idea is quite commonly seen in many studies. But back when we first worked on this, and back then it was actually a pretty cool idea when it was first released. So with that conceptual study done, an engineering concept, we further harnessed its efficiency. We looked into ways of improving its sensitivity against quorum sensing molecule. We improved the way that we can target the Pseudomonas by adding in a biofilm targeting enzyme, because a biofilm is a structure that allows the pathogen to escape or resist antibiotic activity. And then we transfer that design into a probiotic strain called Nestle. Now that we have this in a probiotic and potentially something that you can ingest, we began to test it in a living system to see, does the in vitro activity we observe continue to be true in living system. So we use a very simple model, C. elegans, which is a very standard infection model to use with a straightforward survival result at the end. So we show that engineered probiotic was able to protect the C. elegans by increasing the survival rate significantly than other groups when infected with Pseudomonas. And when we imaged the infection and clearance of the Pseudomonas using fluorescence microscopy, we show that this increased survival rate is perhaps due to enhanced clearance of the pathogen from the gut, because we see a very clean lack of dopamine fluorescence in the probiotic treated group. So we took that as a very promising result, but then it's very a simple organism, so we wanted to use a higher organism. So we used mice infection model instead. So we infected the mice with Pseudomonas and make sure that it's well established. And we subsequently gave our engineered probiotics as a treatment. So upon treatment, we observed a faster clearance of the Pseudomonas compared to other treatment group or the infection control, which actually amounted to a couple of orders of magnitude compared to infection, but about 80% compared to the other control groups. So it showed that our probiotic engineered probiotics was able to clear the Pseudomonas, even if it's actually well established. But we also wondered if it can be used as a protective measure. So if you know that you are going to be exposed or you're in a high risk group of Pseudomonas infection, so if you give our engineered probiotic, would it protect you against the infection? So we reversed the order by giving the mice the probiotics first, and then subsequently challenged with the Pseudomonas infection. So in this case, upon exposure to the Pseudomonas, the engineered one was able to show even faster and greater extent of clearance of the Pseudomonas compared to the other group or the infection group, and which is more than 98% or actually 3 orders of magnitude of the pathogen. So we were able to actually publish this in 2017, very excited about the finding. But as we explored ways to improve engineering approaches in antimicrobial field and one of the concerns that was very soon brought to our attention was the event of antimicrobial resistance. And this resistance is going to be around as long as the tactic or the method we use focuses on killing the pathogenic microbes. So instead of actually direct targeting that uses peptide, so we questioned whether there's a way to get around this. So can we actually target the pathogen without trying to kill the pathogen? So naturally then the question was modified to question, can we modulate the behavior such that their virulence is kept in check instead of killing them. So the question was, we're trying to evolve, like, can we modulate the pathogen behavior? So for this question and another, we've selected Clostridium difficile as our target pathogen. Clostridium difficile, or CD, is quite an interesting pathogen because it has two forms that it can exist. One is in spores form, and the other is the vegetative, the expanding version of the form. And the spore form is quite resistant to antibiotics and to various, it's very tough structure to get rid of. And it may stay dormant, and if you're exposed to it, you may not even be aware of it because it doesn't affect the patients unless there's a disruption to gut microbiome that activates and triggers the CD spores to germinate into vegetative form. And usually this disruption is, it occurs when antibiotic therapy is given. So it's actually kind of hospital acquired in a way. So once it expands and colonizes in the gut, then it actually produces an array of toxin that can result in Clostridium difficile associated diarrhea, or CDAD. And from this point onwards, even if you do recover, so what some of these cells simply form spores and remain and have this cycle going on and on and on. So from this point onward, the CD will just simply shuffle back and forth between spore and vegetative form. And because of this, it renders patients to suffer recurrent CD infection. And this recurrence is about 25%, and it causes extended hospitalization. And it actually grossly affects about more than 500,000 people in the US alone. And it is associated with a considerable death per year as well. Antibiotics aimed at eradicating CD is not a practical or feasible treatment option because you cannot target these spores. And as I understand it, fecal microbial transplant is currently given to CD infection, or CDI patients, and it's pretty much the last line of defense in CDI therapy. So here, if we apply our question earlier, can we modulate the virulence of CD and prevent it from germinating? Can we, sorry, my slides. Yes, there we go. Can we have our engineered probiotic to sense the event of dysbiosis and correspondingly prevent CD germination, and therefore the virulence associated CDI does not take? So we don't even have to kill the pathogen, but we simply have to lock it in its avirulent form. So to build on our quest of this question, we have to understand a little bit about CD. Like, how do they germinate? And why do they, why do they germinate after dysbiosis? So if you can bear with me for a minute, I'll just do a quick crash course about CD biology. So in dysbiosis event, a disruption to microbiome occurs, and this also means a disruption to a metabolism that's mediated by microbiome occurs. And there is a growing evidence that the CD infection and there's a causal relationship between the event of CD infection and disrupted microbiome-mediated bile salt metabolism. So bile salt metabolism that is usually modulated by microbiome is disrupted, and therefore it causes, and it has a causal relationship to the event of CDI. So if I even break that down further, in dysbiotic state, conjugated form of biosalts, such as taurocolate, here, this form, increases, which happens to be a strong germinant of CD. So if this increases, germination occurs, and you have expansion here. Correspondingly, the level of unconjugated biosalts are reduced in a dysbiotic state, but this has been reported to be an inhibitor of CD colonization and growth. So, to sum up, as dysbiosis-mediated biosol metabolism contributes to CDI, we hypothesize that modulation of biosol metabolism can be targeted to disrupt the CDI and fits our quest earlier. So based on this hypothesis, we engineered probiotics that can sense dysbiosis signal, and we have used sialic acid as a proxy signal of the event, and turn on an enzyme that can turn on an expression of an enzyme or biosol hydrolase enzyme that can deconjugate the biosalt. So as a result, we hope to achieve a reduction in the taurocolate of the conjugated, an increase in the unconjugated form to inhibit germination and inhibit growth as a result. And we hope to achieve hydrolase mediated modulation of biosalts. So I hope that is a clear explanation of our basis and the hypothesis. And therefore, so we searched and identified and characterized various enzymes that can do biosol modulation, primarily through deconjugation. So we identify, and our recombinant enzyme have shown that it can efficiently convert taurocolate to colate, almost 100% efficiency. And this enzymatic conversion of taurocolate to colate can significantly reduce and inhibit the germination of the C. difficile and the spore germination. So taurocolate alone can cause this level of germination. And when the enzyme is there to convert the taurocolate, we no longer see the germination of the C. diff. And the last figure shows that this biosol modulation, the taurocolate to colate, now the colate can have an effect on the cell growth. So it significantly reduced the growth of the C. difficile vegetative cells as well. So having enzymatic modulation resulted in inhibition of the germination of endospores, and we managed to show that growth was also inhibited, growth of the vegetative cells was also inhibited. Next, we engineered the system called sensor amplifier and actuator system to modulate the biosol to develop engineered microbes that inhibit C. difficile infection. So we basically placed the enzyme expression under the sensing module, which is aimed to detect sialic acid, which is a proxy signal. And this is chosen because there's a report showing that the sialic acid is significantly elevated during microbiome dysbiosis. And also, it's also implicated in a C. diff activation as well, and some signaling. So when we actually first extracted the sensor, even though dynamic range of response to sialic acid was good, but the level of response was quite poor. So we had to add a transcription factor-based genetic amplifier to increase the level of expression of actuator enzyme. So this figure shows how the steps that we have taken to gradually increase our activation response to sialic acid. And at the end, this is the genetic circuit of sensor amplifier actuator system that enabled our probiotic to be dysbiosis responsive modulatory of bile salt profiles. So once we had the system in probiotic in place, we tested it against C. diff in vitro. So we showed that our engineered microbes can indeed reduce the germination of C. difficile endospores. And when it's co-cultured together with vegetative cells of the C. difficile, it pretty much rapidly reduced the growth of the cells within 6 to 12 hours and maintained its inhibition. So we also confirmed that our engineered microbes in exposure to the human gut epithelial cells, that the toxicity that can be observed by the C. difficile is greatly reversed in engineered probiotic presence. Of course, this is all in vitro, so we wanted a more conclusive results in vivo. So we established a C. difficile infection model in mice and tested the efficacy of the engineered probiotics. So the figure A shows the protocol that we have used by giving antibiotic first, probiotic, followed by C. diff, and tracking the progress subsequently. And the figure B shows that the mice with the engineered microbes led to 100% of survival rate, while the survival rates of the control groups range from about 15 to 60%. And figure C shows that the mice with the engineered microbes had a lower clinical sickness score, which indicates mild symptoms, while other control groups had score ranging from 8 to 10, which indicates moderate to severe symptoms. And I just want to highlight that the clinical sickness score is a combination of the quantitative matrix based on the activeness of the mice, the body weight loss, and the fecal stool consistency scoring. So based on these three combined, you have a total of 12. So overall, our results suggest that the microbes engineered to modulate bile salt metabolites can inhibit C. difficile infection in animal models. To see the metabolites changes in these mice, in other words, we had to ensure that our microbes were activated in the presence of the salicylic acid, and we had also had to ensure that it modulated the bile salt as we intended. We looked into the level of salicylic acid after the antibiotic treatment, as well as looking at the tyrosine and cholate level afterwards. So figure A also shows how we collected for what purpose of the evaluation. Subsequent figure shows that salicylic acid level indeed increased compared to the start of the antibiotic and end of the antibiotic treatment. Even within 1 to 2 days, you will see a significant increase already. And this set of figures shows that post-probiotic administration, you begin to see a great reduction in tyrosine in these mice, which corresponds to increased level of cholate, the deconjugated product by the enzyme in these mice. So overall, our results suggest that the microbes engineered to modulate the level of intestinal metabolite, the bile salts, do support the inhibition of the C. difficile infection results that we observed in animal models. Right. So in the remaining time, I would like to introduce our next set of strategies that we're trying to explore in our lab is from modulating a target cell behavior. We wanted to sort of move into how we can use our microbe to modulate the cell interaction that occurs between the microbes and the host cells. To this, I would like to introduce our work against cancer that we have done a few years ago. So our idea started with the idea of how can we harness what we eat to our health advantage. And we looked into this particular group of chemicals that can be found in cruciferous plants called glucosinolates. And we realized that this is actually one enzymatic conversional way to forming sulforaphane, which is an active anti-cancer compound. And this conversion is mediated by an enzyme called myrosinase, but this is it's not found in abundance in human gut or human system. So our idea was to have our probiotic engine to deliver this enzyme so we can unlock the glucosinolate that we consume into an active anti-cancer compound. And we also wanted to have this kind of conversion in a close proximity to where the cancer is. So the idea was, can we get our probiotics to bind to the surface of the cancer and then convert the glucosinolates into sulforaphane to kill the cancer cells. And once the cancer cells are killed, then it will actually have released the probiotics. It will no longer have anywhere to bind to augment clearance. So in order to have this design to work, we need to actually have three things. First, we needed that active enzyme that will allow the conversion. Second, we need a mechanism to have a cancer localization. And also the third is, most importantly, it had to work together. So these are the three sets of results to answer those three things. So we first looked into enzyme optimization. So we screened a panel of myrosinase enzyme group, and we had to look for those that had highest activity in the physiologically relevant conditions. For instance, during the pH that is physiological and at 37 degrees, which is the body temperature. So we have selected this I1, which is the myrosinase enzyme. And also, in order to actually augment cancer binding or localization, we looked into the surface of the colorectal cancer, and we noticed that they would have this layer called heparin sulfate, SSPG. And a bacterial protein called HLPA has a great affinity to this group. So by expressing the HLPA on the surface of our E. coli, we managed to actually have a very specific binding to colorectal cancer cell lines, which is actually not so much observed in the gastric or other cell lines. So it actually showed a very specific binding affinity to colorectal cancer cells. So by putting them together, we tested in a cancer model where we had given the carcinogen and DSS, so it's an AZM DSS model over the period of 14 weeks. Then we gave a weekly administration of the probiotic. And when the probiotic was given, and with actual broccoli, we fed mice broccoli, we see a great attenuation of tumor formation. And actually, even if they do form, it was actually much smaller compared to any other groups. And we've shown the highest localization of cancer because when the enzyme and the HLPA was co-expressed, it was found highest together with the colorectal cancer tissue. So greatest tumor prevention was observed when fully engineered Nisul with broccoli supplements were given. We are currently working on improving this to have it translating this idea further. And with that, we are also actually looking in various other types of cancer. So with the evolving strategy that employs modulating cell-to-cell interactions of various forms, we are looking into a wider range of human diseases. There are a few nearing completion and few in testing POC, the proof of concept stage, and some are in preclinical testing stage. So soon, I hope to have another opportunity to share the other works, exciting works with you. But with this, I would like to end. And this is actually Matthew's lab and together with my team. And these are the various funding sources for the research center to support our research in synthetic biology. And thank you again for having me in this SynBYSS conference and giving the opportunity to present and be named right there with Sang-Hyuk Lee is actually quite an honor, actually. And I look forward to working with you, many of you, to advance synthetic biology in future and as well as in Asia. Thank you very much.
Thank you so much. I mean, amazing talk and also very important topic. So I really love it. And also, you know, your and Matthew's group is always one step ahead. I'm glad, again, not to seriously proceed further my biosort related project. Actually, several years ago, after some failed experiment and after seeing Chris Boy's biosensor, bioacid sensor, I mean, a few years ago. So that's amazing, amazing. Actually, I have a question, but I wanted to wait. I have the one chat. That's about thanks, new insight. Oh, that's not question. So, Bill, do you have any question or challenging or comment? Anything?
No, it's wonderful talk, and thanks for letting me be part of this as well.
Oh, thank you. So let me check in the case of any Q&A, question from audio. Okay, I'll ask question. So I have a very challenging question, so be ready.
Cool.
So I always wonder why pathogens exist in terms of evolution perspectives. So they make their host sick, basically, and is it beneficial for their survival? And that's kind of a question. And if the host dies, there would not be any added fitness benefit, I believe. And obviously, bad humans exist harming others, but why have small bacteria evolved to harm the host? Now, I basically understand if pathogens are killing other competing bacteria by toxin, whatever, that would make complete sense, but harming the host itself does not make sense to me. So could you kind of share your perspective?
I actually had some sort of discussion on this before. My take is what we term as virulence is actually a very self-centered point of view. So what we view as virulence for pathogen, they're opportunistic in a way. They are just trying to grow and occupy a niche, and in doing so, and whatever competitive edge it provides them, some are just how they survive. But it's just that our body responds and reacts differently, and it ends up being what we deem as virulence. But in microbial perspective, personally, I think they're just growing and adapting and trying to sort of compete and trying to sort of occupy that space. And when the opportunity arises, like dysbiosis, when there's literally for them, that means more space for them to grow. But because they're So in their normal number, our body has adapted or know that that's actually normal level. But all of a sudden, when there's too much of them, I don't think our body is used to it. So we kind of mount the immune responses and trying to sort of get rid of it and actually get sick. And as any pathogen goes, it derives no benefit killing the host. So usually those that after outburst, and hopefully, unless it's very notoriously, in the case of C. diff, because it's spores, that's actually their mechanism of survival, but it works very bad to our advantage because it means we can never get rid of it fully. So I think it's just difference of understanding that's just gone wrong in when our health is compromised. So when we are healthy, we can actually keep them in check.
Oh,
So that's my personal perspective.
No, I love it. I mean, actually, that makes complete sense now because I mean, we need to kind of change our perspective, you know, from the bacterial perspective, right? So that's kind of complete makes sense. So thank you. That's interesting. So now I see 11 o'clock my time. So actually, I wanted to kind of close. And also, I also want to comment because your early Singapore photos make me want to visit there again. In fact, Matthew Chang is chairing and hosting the Metabolic Engineering 15, and actually I am co-chairing the conference as well next year. So I will be there anyway. And then stay tuned. I mean, we will have the more information about the conference. So okay, I want to close. So thank all for joining and staying today, especially our speaker, you know, now midnight, I guess. And we'll meet again next week on April 28th, Thursday, the same time, the same Zoom link. We'll have the Professor Ed Boyden at MIT on HHMI investigator and Professor Han Jiao at Rice University. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with today's speakers and me, and I will promote you to panelists who can speak and show your handsome and pretty face. So thanks everybody, and I stop recording. Let's give me 1 second. Okay.